Okay, so a few things. First of all, we missed the lecture, so that changes a little bit the schedule. But we have two options. I'm going to let you choose. Probably you're going to choose the option of not presenting, but hopefully there's some people that are interested. So there's 4 topics left for me to talk about, okay? Microbiome, gene therapy, prostate cancer, and Neanderthal, right? So today we're going to cover prostate cancer. So we're just going to continue with the flow that was there before, but that means we'll start presenting right after the microbiome lecture, I think, right? So that leaves the Neanderthal talk. Now I have 2 options. Either I record it, you guys listen to it, and that's it. I can also not record it, not include it, and you guys don't listen to it. Or I can record it, but then not include it in the exam. That's probably not a... Yeah, obviously remove stuff from the exam might make you think that it's going to be easier, but this paper you read it many times, so maybe that's an advantage for you. So think about this. I guess we can vote on this. So do you guys want the lecture but not included, want it, and want it to be included on the final, or don't want it at all? Let's start with the option of lecture but not included on the final. How many people? Okay, so 5, 6, 7. 7, okay. Include it on the exam. 1, 2, 3, 4, 5, 6. Mark, you're cheating, no? Okay, so another last option, just don't care about it. That's it, Maria, Team Maria. Okay, so I will still record it. I will post it. You can listen to it, but I will not include it on the final exam, okay? Bad choice, guys. Could have been an easy question that you already practiced, you know? But it's okay. The exam is pretty simple, pretty easy. It's like less, it's easier than the midterm, okay? There's more direct questions, let's say, because there was a little request, so I put it in there, okay? There's 7 questions for the final exam, so you have plenty of time to answer that. I'm saying that because I already submitted the exam, so I already know what's in it. It's more learning. There's a little bit of thinking, but mostly knowing what the hell we've been talking about, okay? So I'll just keep it at that. So you have 3 hours to do 7 questions. I think you can reinvent the whole course if you want, okay? So that's that. The other stuff is I added a few things. You saw the notifications. I put the presentation papers here. That's the first thing, so you can read them. You can read them, not read them, whatever you want. I suggest reading them because maybe they are in the final exam, or at least small questions about them. So obviously there's that. The presentations, I will have this rubric for each student every time printed. You can take it and then answer here. This will count towards, you basically grade each presentation. This does not count for the team's grade, but counts for your own participation marks, okay? So the reason why I'm doing this exercise is for you to actually focus a little bit, because we tend to fall asleep in presentations. That's the first thing. And I need you to learn the material, but most importantly, you'll be asking a question per presentation, okay? So you just write it here and put the right answer, wrong answers. It's like a multiple choice that you're creating, okay? And this counts for your participation marks. Now, obviously, I'll be using the exact same one to evaluate each presentation. So if you're asking yourself what the hell is expected in the presentations, this is it. Okay, so that's that. One thing that I have to say in advance, we need to have a discussion time after each presentation, so try to keep it at 25 minutes. If it goes a little bit beyond, I might have to stop you, and we don't want to do that, okay? One very easy trick for presentations is some people like to write script. I don't know if, I used to do that when I was studying. I used to write the full script. That's not the best way of working, okay? I realized that with time, because the moment you forget something in your script, you block in the middle of the presentation. So instead, if I can give you only one trick to kill this, is to actually know the paper so well that you don't even need to have a script. You could just take a look at the graphs and describe them, okay? Instead of writing a script, slide 1, I will say this, slide 2, I will say this, and then you sound robotic, you lose a lot of points, okay? So try that. If that doesn't work for you and you prefer scripts, go for it, whatever works for you. This is just my trick. Does this make sense for the rubric? Okay. The essays that so assignment number 3 is corrected. The grades, I had to review it. Some people talked about Morgan Hunt. So that was kind of accepted as an answer for mapping of a gene to an autosome. The thing is, Morgan Hunt's experiments were not really mapping in absolute terms, in the sense that you didn't know which chromosome exactly they were on. So they were treating them as first, second, and third chromosome, but they were really groups of genes together that were said, these two are close to each other, so they're on the same chromosome. But it's not that exact chromosome. So the earliest ones that I've seen in the lab, I'm still in the lab, in the class, there was the ABO group, the Duffy group. The Duffy was first, ABO was second. I accepted both. The reason why you cannot, so Morgan's experiments are like the basis of that question, okay? The thing is, to be able to actually map on a chromosome, the question that I asked was also in the lecture related to karyotyping and staining, right? So there was a link to do there. So one of the things you need to be able to map on a chromosome at that time before sequencing was staining and seeing abnormalities based on the variance in the population. So a lot of people that got that right, I had to reevaluate, because before I was not accepting even like Morgan Hunt at all, but you know, being nice. The other thing is I'm gonna regrade your, because I feel like I was a little bit harsh on you guys. I went home and I was like, shit, I messed up, but it's okay. I will regrade your assignment number 4, but the only thing I'm changing is the dystrophin gene, okay? Now, I'm doing this because most of the class got the dystrophin gene, but you have to also take into account what I'm telling you now, and just, you know, think about this in the future. So if you go and find dystrophin gene on there, and there's thousands of papers that talk about the dystrophin gene, your job as a scientist is to verify that information. Don't just put it there, okay? And because the paper, if you see it in 1995, for sure it changed. Like if in 1 week, maybe my research will get scooped, right? What I'm doing right now. So you have to verify this stuff. And usually when you're a student, you don't think that you have the skills to actually verify this stuff, but you do. So just go on the databases and make a filter bigger than 2,200,000, whatever, and see if there's a new one that appears in there, okay? So that's that. The idea is not to penalize you, but to little bit of a lesson here, okay? One thing that I'm not happy though, but I've been correcting your essays. I'm almost done. There's the discussion that's left, and then I have to recount all your points. So the way I do it is I correct the intro for everyone, then the methods for everyone, then the results for everyone, and I keep it like that so I don't forget what the hell I was saying for other students, and then treat this one differently from that, okay? One issue that I've seen, first, certain people didn't apply themselves very well. I would love to say it in a different way, but you don't change citation style in the middle, like right after like 2 times, then you switch the style in the middle. Why would you do that, right? And so that then makes me think, was that sentence copied from somewhere else, okay? That's the first thing. And then if you go on the paper, you find something similar in there, but you don't do anything about it, okay? So that happened. The second thing is, you have ChatGPT, correct the freaking thing on there. Like, ask it to correct it. Write it, but then ask it to correct it so that you don't lose half of your points on the style here, like this part here, okay? In terms of introduction, most of you had it right. You talked, the first sentence that most people had was, prostate cancer is the second most prevalent cancer in men in the United States. You guys know, right? You had the same sentence, okay? Now, one person had a reference to that. everyone else did not have a reference. And the reference, it's Sarah Borham that had that reference in her thing. Like, no, I didn't know that, prostate cancer was the second one most prevalent. You guys knew? Of course, come on, man. Right? And then you're talking about things one after the other, and you're pulling out statistics, but no reference. What the hell is happening? Right? I mean, sometimes you don't think about it, so that's all right. But for this one here, I'm being super nice on the referencing, okay? Because most of it is not common knowledge, most of it. So that's the thing. The length, top length of the paper, 9 pages. Do you know who it is? Sarah again. And then we had 2 more papers that are like 8 pages and 7. Guys, keep it to 5. You're killing me. Keep it to 5, okay? I don't have any problem with that. It's okay. I can read it. The problem is that if I say to people 4 to 5 pages and then one writes 9, that person tends to cover a lot more than the other, and they get more space to talk. So it's more space to provide the information and have a flow, right? So then that's kind of penalizing the others. Okay, so for this one, really keep it to 5 pages max. Okay, the first one, it's okay. I'll act like it was not there. I'll read the extra 4 pages. Okay, I had one paper that had 1 reference. One paper, 1 reference. How? That's not normal. Okay, that's not normal at all. It's telling me that basically either the person assumes that everyone knows everything that's written in there, which is not true, because I learned a few things in here, in this paper here. So if I'm learning them, I'm pretty sure there's people that are learning also, okay? Now, what else? One thing that was missed is did you guys follow this? Okay, honestly, not like disrespecting or whatever, but I feel like some people didn't even look at this and just wrote. Some people. Most people, they had their stuff, but if you see statistical analyses are interpreted and explained, that means if you want 9 or 10, you have to put those statistics, right? And so most people forgot about the statistics. Limitations, this one I'm ignoring because I feel like at this level it's a bit hard to find limitations. But today I will show you 2 limitations, and it's going to click in your head like, oh shit, this is obvious, right? It's just, you don't think of them as you go through it because it seems very complex as a paper, okay? As a matter of fact, because I'm talking about this, I'm going to present the two limitations and then go back to the paper, okay? So the first limitation that you find is the fact that they had to test those genes that they found in vivo, right? When you're talking about metastatic aggressive cancer, that means the cancer is in a microenvironment that allowed it to detach and then move to somewhere else. Now, if you're taking cells from outside that are doxycycline inducible, those cells you modified by yourself and then you inject them in the mice, you don't have the microenvironment. What you have is cells that were cultured in the lab, modified in the lab, and then injected in the mice. And that is not the actual cancer itself, right? So this is one of the limitations of the study, is that their tests to make sure that those are actually cancer-causing genes in vivo is not fully there, right? Because it's not really representative of the actual microenvironment from where the cancer comes out. Obviously, if you mess up the cell and you put it somewhere in the mice, most of the time you'll get something, right? So this is one of the limitations, which is kind of obvious, but you have to think about it, right? And I could go on with the 2 million cells that were injected and things like this, right? Obviously, if you put cells in a space where there's nutrients, they will grow. And if the cells are not supposed to be there, they will grow on top of each other. So is your tumor really coming from actually kinesin factor 4A, or is it just the cells developing in there? That's the first thing. The second thing, they were talking about the filtering system that they had with the clinical data set. Now, that filtering system is taking data from their CRISPR screen and comparing it to actually clinical patients. But how can we say that prostate cancer is potentially caused by KIP4A if their filtering comes from a cohort that is not evenly distributed. So you know that they had those two papers, the Quagley or whatever, and the other name there, okay? So if you open that study and you look at the demographics of the patients from where they got the DNA samples and analyzed the factors, you'll see that there's a filtering bias. KIF4A is true potentially for white male patients, but probably not Asian, Black, or whatever, okay? And if you take a look at the secondary site of metastasis, you'll see that it's tilting towards bone and lymph nodes. So cancer, as I will explain it in a few minutes, is kind of complex. It's not just like that. And prostate cancer is more prevalent in African American men. So actually this data here is relevant, but it's not as relevant because the major demographics that have this cancer are African Americans. So this potentially does not even apply to them, right? So this is kind of the limitation of their filtering system, okay? Obviously, we're talking about androgen deprivation therapy, right? And we do this through these two drugs here. So you reach androgen deprivation therapy, you reach castration resistance by doing the treatment, right? So you do the treatment to try to cure it, and then it becomes resistant to the treatment. But in this data set here, those patients, most of them were not treated with the two drugs. So it's not even representative of actually people that get prostate cancer treated with ADT and then resistant. Does that make sense? So these are things that you can look for. If you ever have something clinical, just go at the cohort. This is directly for sure you'll have a bias there. Like just go look there straight. You'll know that the population, obviously if it's done in US, you're not gonna find the biggest population here that was tested is Algerians, you know. So there's always a filtering bias in those data sets because they just take whatever possible patient they can find and agrees. And if you're in Algeria, most of them will be Algerian, but if you're in the US, most of them probably will be white, okay? So that's that. So, give me 1 second. Let me just close this. So in your writing, the limitations were hard to find. Some people actually had some interesting ideas, so I gave points there, but if you didn't mention it, I didn't remove points. So this is just bonus, okay? What else? What else? Other than that, the rest is pretty good. So you should get the answers, the grades by tomorrow night. What the hell am I doing? I'm gonna finish correcting the discussion, and then I have to just go through them all, and then count the points and all that. So that should be there tomorrow. Then it gives you about a week and something to do the corrections. Okay? Please put the corrections. You get a better grade. I get an easier time correcting you. So implement everything, and then from there, you should be good. Okay? Cool. Questions? Okay, so 3 presentations today. 4th one that will be recorded but not included in the exam. I'm going to show you a little bit how to kind of present your stuff, the papers. So I'll be presenting 4 papers in a row. Sometimes I'll focus more on the results, sometimes more on the background, sometimes more on things so that you have an idea, okay? And you can use this as an example. So try to do this fast as usual. Today I'm going to cover the paper that was assigned to you for prostate cancer, okay? So usually the way you start your presentations is by giving some sort of background, right? Most people like to talk about disease and death. I try to switch it up a little bit, okay? So you can also go the route of just talking about cancer and the different types and then go on from there, okay? So I have a question for you guys. Do you think that there are cancers that are transmitted from one generation to the other? Yep.
Absolutely. There can be an imbalance in the way apoptosis is regulated in the cells, and that can be transmissible genetically from parent to children.
Yeah, so that's a very good point. There's one example of this type of cancer that is transmitted from generation to generation, and probably the one that you know, the BRCA genes, and then the mutations in them that are passed from mother to daughter in terms of breast cancer. There are cancers that are not genetic, though, that are not inherited, I mean. Can we call those genetic diseases?
They're still caused by a genetic error, so I guess you could say that,
Yeah.
yes.
So cancer, at the end of the day, comes back to genetics. Okay? So you can have it caused by food, smoking, drugs, whatever the hell you want. At the end of the day, it will start playing with your DNA because it has to change some stuff in there that allows it to proliferate and do whatever it has to do. So if you read on cancer, you'll always be reading about these hallmarks. You guys know about this? So there are 10 of them. This one here is supposed to be number 4, not 6. This is 6. But basically, there are 10 hallmarks that determine if something is a cancer or not. And there's the same thing for aging, for example. Okay, the 12 hallmarks of aging. But mainly all these, so invasion and metastasis, this is a change in transcription of some genes that are important for movement, inflammation. So all these basically come back to one thing, which is your genes, okay? And the problem with cancers is that it's not one disease. It's many smaller diseases within the frame of one big umbrella term. And so what happens is that the way we're treating the cancers today, in maybe 1,000 years, it won't be valid potentially. Why? This is why cancer is crazy. It's because it's able to reprogram itself and become more efficient at evading the treatment that you give it. And so sometimes the question is, do you treat or not? Because if you treat, then you can accelerate, for example, the death of the patient or the person that's getting treated, right? One thing that is very interesting that most people don't know, usually people don't die of the cancer, they die of the treatment, okay? Why? Because what happens is that you get the cancer. If it's treatable, for example, the simple breast cancers that are responsive to estrogen therapy or whatever, those you'll be okay. And then you get checked up for 10 years until potentially it comes back. But if you get a cancer that has no drug for it, you usually end up doing either radiotherapy or chemotherapy. And what happens with those is that they destroy your body. Like when I say destroy it, they destroy the cells that divide the fastest in your body, right? And those are usually your lymphatic system, your immune system. And most of the time what happens is that you'll die of other diseases like sepsis, infections, and things like this, okay? The cancer itself, for it to get to the point where it kills you, it will require a lot, unless it's like a brain cancer or blood cancer or something like this, you know? But if it's an epithelial cancer, you'll have to wait for it to metastasize and go somewhere else that is viral, and then it will end up killing you at some point. But usually it's the treatment that does the killing, okay? So there's a need for developing therapies that avoid all this stuff, right? And for us to be able to find these kind of drugs that avoid chemotherapy, which is a generalized treatment, you need to do high throughput assays, okay? So you can't just go like in 1960 and try to go gene by gene and figure out your whole career if that gene causes cancer or not, right? The best way to do it, you have to kind of have a technology that is able to catch up with the speed at which the cancer changes, okay? And they acquire mutations like it's nothing. Like you can have like a patient treated for a year, and in that year it will become resistant 4 to 6 times to different drugs, right? At some point you have to take the decision of stopping the treatment or continuing and potentially killing the person. Usually people just stop and go in palliative care, okay? So because this cancer evolved very quickly, then you end up having to kind of find the targets quickly and treat them quickly, right? And so this is why high throughput techniques are very important, and this is why I presented this paper to you, okay? So the idea is that you try to identify genes that are involved specifically in that cancer at that time, right? And then you try to establish its link to it being clinical or just, a random finding. And this paper that I'm presenting to you today that you wrote about does exactly that. Now, if we're able to speed up this thing here and get it into the 1 week kind of span of time, we could help a lot of people, okay? We have ways to kill the cells, but we don't know how to target them, right? This is the problem, but we can kill the cells, okay? I know you guys probably all know what prostate cancer is, but just to get back to it, okay? So prostate cancer is a disease that affects the prostate. The prostate is a male sex organ that produces a white liquid that mixes up with sperm, okay? So that liquid, forget about the liquid, more the actual prostate, is responsive to testosterone, okay? And this testosterone with time will lead to the prostate enlarging, okay? This is why most men after 50 years old, they have to do these tests to make sure that their prostate is not enlarged, because this is a high risk, okay? There are a few causes for this, and there are a few differences between the demographics for that affect prostate cancer, and you'll see the differences in the next slide. But one very important thing that is interesting to understand is that these cancers they're easily treatable, but they become super aggressive, okay? So what happens is that if they become resistant, then you end up pretty much having a lethal cancer, okay? And there are a few risk factors for this, which probably will sound weird, but you'll understand why, okay? So obviously, ethnicity or race, some people use this term in scientific literature, but ethnicity, your origins, dictate if you are at higher risk or not, okay? The reason why white men are more at risk than Mediterranean men, for example, it's because of one thing, the diet, okay? The diet in America is full of red meat, and red meat contains a lot of hormones, okay? So that's the first thing. If you have too much testosterone in your meat, what happens? Well, you end up stimulating that kind of cancer, okay? The other thing is Mediterranean food is less rich into red meat and richer in fish and vegetables, and so they have a lower risk. That's one thing. Second thing, most cases occur at the age of 50 and above, and this is mostly due to the fact of the mutations that get accumulated in the receptors and all that, okay? Interestingly, BRCA mutations were linked to breast cancer, but now they're starting to appear to be linked to prostate cancer. So this is what I was telling you about the plasticity of cancer. It's just a mess, right? Like at some point it's not, but then it becomes associated to BRCA mutations, which is weird because before it was not, right? So these cancers are evolving and using different ways to proliferate, right? And so maybe in 100 years, this will not be relevant anymore, and maybe prostate cancer will not even be linked to androgen receptor. Some crazy statement there, but you understand the idea behind this, right? One good thing about cancer is that as we develop drugs for different cancers, we can then later repurpose them. So we can start treating these as this type, right? This type of breast cancer, but obviously prostate, breast, different organs, but we can try to take something that we already know and then reuse somewhere else, okay? Just a few types of cancers. Some of them are interesting, you probably didn't know. So there are a few sex-influenced cancers caused by different things. The one that we're talking about today is caused by mostly environment as well as hormones. But there are other types of cancers that are caused mostly by environment. For example, this mesothelioma, which is caused by asbestos inhalation. Now, how come this is a sex-influenced cancer? Well, if you look at how time went, men usually are the ones that are working close to asbestos, right? So what happens is that they get this cancer through working into buildings or whatever and coming into contact with asbestos. Asbestos will infect your lungs on the pleura, so that's the exterior of the lungs. And then obviously if you come home, so I'm talking a long time ago, now everyone is working, man, women, doesn't matter. But at the time, if you come home with your dirty clothes and then your kids and wife actually get into contact, then you have a secondary contamination. Hormonal cancers, those are obvious. They're caused by androgen receptor for the prostate. Breast cancer, depends what it is, estrogen, things like this. One that is very interesting. So if you remember last lecture or lecture before, I talked about the different numbers of X and Y chromosomes. So if you have two X chromosomes and one Y chromosome, you end up being phenotypically male, and that's called the Klinefelter syndrome. And these people are more prone to breast cancer. So I'm talking about men but more prone to breast cancer because of the high levels of estrogens. Okay. Now I don't want to do politics, and I don't want to get into trouble, but I will say this from a scientific perspective. Canadian government allows for a gender reaffirming surgery and hormone treatment. And one of the things that these treatments do is that they switch up the hormones in your body, knowing that medically they will cause cancer. But obviously, kind of seems weird that the doctors allow it, but I understand from where it's coming. It's coming from the fact that those group of people that usually undergo these reaffirming surgeries and hormonal treatments are vulnerable, and so to make their life better, mostly psychologically, you have to take the least of the two, right? And it seems to be working in terms of decrease in suicide rates, okay? But one thing that is important to understanding these things, the change in hormones, the treatment with actually GnRH and estrogen, testosterone, and all that stuff will lead to higher rates of cancer, okay? And this we know for a long time, just from kind of this example, okay? So goods and bads, taking the least bad of the two options, okay? Now I'm saying this from a scientific perspective. I don't care about the other stuff, so take it just as that, okay? Good. So back to the paper. So in this paper, they talk about a model, and the model is the aggressive prostate cancer, the metastatic castration-resistant prostate cancer. And typically, these cancers are initially responsive to androgen removal therapy, and you do this by using things like enzalutamide, which will compete with the testosterone or dihydrotestosterone and bind to the androgen receptor and prevent it from going to the nucleus, right? So this is how you do the androgen removal. You don't actually go in the person and remove all their androgen. That does not work, okay? They're androgens. Obviously, androgen is a term that englobes testosterone and dihydrotestosterone. But with time, what happens is that the cancer is way too smart. It becomes resistant to this therapy here by acquiring mutations that make it not require a ligand. So it does not require testosterone or dihydrotestosterone anymore, and then just doesn't care about the androgen removal method that you're using. Now these people, unfortunately, the cancer becomes more aggressive because their androgen receptors are always active. A subtype of them, there are some, it's a bit different, the mechanism, but most of them, the androgen receptor is always active, and so they end up passing away, okay? In this case, in this paper, they were trying to figure out what genes were androgen independent so that they can kind of find new targets. That would be the targets that kind of get overexpressed when the cancer becomes metastatically castration resistant. So it does not care about androgen anymore. Things change in the cell, and so you want a new target other than the androgen, right? And so they used the CRISPR-I screen. They looked for phenotypes that led to a basically decrease in amount of cells, okay? And then they look at the top genes that came up. So CRISPR-I, you guys know it because you read about it, but very quickly, it's the same as CRISPR. The only difference is that you have a Cas9 enzyme, which is supposed to cut the DNA, does not cut it anymore. So that's your dead Cas9. It's bound to the single guide RNA that targets a certain region of the DNA. The only additional thing that you have in CRISPR-I is the fusion of the Cas9 to a repressor protein. And that repressor protein, we learned about it when we talked about transposons, if you remember. It's called CRAB. CRAB is short for croppo-associated box. And usually what CRAB does is that it will repress a target gene. So you will bring the Cas9 with the guide RNA close to the target gene, and then CRAB will do its job of repressing this region here. Do you remember this figure? Something that looks like this. You have CRAB. CRAB brings CAP1, and then I told you that it either methylates or acetylates the DNA, right? So this is your CRISPR-I technique, and it's based on the mixture of this Cas9 that is dead with the CRAB. If we're talking about CRISPR-A, what would be the difference? Anybody? Yeah. Sorry?
It's the activation, so promote the expression of the target gene.
How would you do that?
Enhancer of some variety.
Very close. So what you would do instead is that instead of the crab, you would have transcription factors, right? So it's the idea behind the technique is the link between the two proteins, and then the protein that is here then recruits everyone else. Okay, so you'd have something similar to this, but instead of crab, you would have, I don't know, estrogen receptor, for example, and then the estrogen receptor will bring all the other different proteins that are necessary. So you have your CRISPR screen. You have now to build your library and put it into the cell lines. And the way you do this is by building tons of plasmids. And each plasmid has a single guide RNA in it. It also, sorry, not single guide RNA. It has a Cas9. So there's two things. There's the Cas9 blue fluorescent protein CRAB module, which is just a plasmid that you'll use a lentivirus to transduce in the cells. Okay? Blue fluorescent protein is just to filter out using the flow cytometry technique. So whenever the cells that have this blue fluorescent protein pass by, they get sorted to the side because that means they have an insertion of this plasmid. Okay? Now, if you were interested in this exact specific plasmid, you can actually find it online because people post their plasmids and then you can buy them. So this is from the Gilbert lab, which is basically the person here that ran this study. And this is the plasmid that they have. So you can go take a look at it on AdGene. If you ever read on a plasmid somewhere else and you're interested in that specific plasmid, you can probably find it on AdGene. Okay? Then you have to design single guide RNAs. And if you're trying to target all or most of the genes in the genome and you're using 5 to 10, then you end up designing a lot of single guide RNAs. I'm not sure how they did this. Probably computationally, and then just send them for, I keep saying printing, but it's not printing, it's just synthesis, right? They just synthesize the guide RNAs and they just bought all of them and just ran a full-blown study, okay? So before I get into the details, I talk in the assay guidelines to talk about the controls. So there were a few controls. Some people mentioned them, others didn't. But the controls, there were many in here that you could talk about, okay? First of all, they used 10 or 12 cell lines. Some of them are prostate cancer, some of them are not. So this is one of the controls that were used. And then you had benign and malignant, androgen resistance, androgen receptor dependent and independent. So these are all different controls that were used. So it would have been nice if you actually discussed this because they are important controls, right? The second control that they used is these. They tried to shut down, because you made this CRISPR, they made this plasmid. So you made this plasmid, you have to test it before you start just, destroying everything in like 200,000 cells, 200,000 different genetic backgrounds. So what you do is that you're going to select a few genes that you know are already expressed in your cells. You'll try your system on them, and then you'll extract the mRNA and see if the levels of that transcript decreased, right? And they looked at a few of them, and they see that, yes, their system does work. So they designed single guide RNAs that target DPH2, whatever, okay? And C means non-targeting control. And then they see that the non-targeting control, for example, does not change the transcription levels of that gene, while the single guide RNA that they created does work. So their CRISPR-I screen does work, and it works in all the cell lines, okay? This is an important experiment. Now, the framework of the study. Okay, you know that your system that you developed works. Now you need to kind of think about your study, right? So the idea is that your goal, you want to look for genes that, when inactivated, will lead to a decrease in cell proliferation, maybe even death, okay? So that means that if it's active, it causes higher proliferation and leads to higher levels of oncogenesis, let's say. When you identify those, obviously some of them will be related to prostate cancer, but most of them will not be. And so you need to confirm if they actually are prostate cancer specific or not. And for this, you need, for step number 3, you need clinical samples. Obviously, as you saw right now, the clinical samples are biased. So I can say that they were actually trying to hide this thing here, because in the way they wrote their text, they're not saying our data contains only like 90% white male, right? They were just prostate cancer in general. And I don't know why the reviewers didn't spot that, but that's like a little bit of bullshit, okay? So this is one of the bias they have. But you need to use clinical samples. So how do you do this? How do you do this? Obviously, you guys know, but let's act like you don't, okay? So you're going to take cells. You're going to put in them the plasmid that contains your, what do you call it? Your Cas9, your dead Cas9 with crab protein bound to it. And that will, the lentivirus is just to create stable cell lines. So it's different from transfection. Transfection, you would basically use lipid nanoparticles that just put the plasmid in there, but the plasmid gets degraded. Lentivirus, what it does is that it has that cycle where it integrates the DNA inside the genome of the infected cell, right? And so then you have a stable expression of that plasmid, right? So you'll use lentivirus and then add to it all the single guide RNAs, and then just infect at random cells, okay? Antibiotic selection, this very simple. Your plasmid contains a selection marker for, usually when it's human cells, your mammalian cells, human cells, you'll use puromycin. This does not change for anything, okay? And then you'll get only cells that are expressing your different single guide RNA, okay? Each cell has a different one. And then you collect at t equals 0, extract the single guide RNAs, and then sequence them, and then do the same thing 8 days later. One thing that is weird, some people in the class did not even talk about extracting the single guide RNAs and sequencing them. They just went from CRISPR screen to 1,472 genes. What? The most important part is actually the sequencing and the idea behind the sequencing, right? And so usually when you're talking about the methods, it's what did you measure, how did you measure it, and why did you measure it, right? And if you're not telling me what you measured and how you measured it, weird, okay? But it's okay. So then you will compare the amount of single guide RNA at day 8 and day 0, which is your basal level, and see what's the difference, okay? And I'll get back to this in a little bit, but the change between the single guide RNA levels will tell you which genes are cancer causing and which ones are not, okay? So the library had 200,000 single guide RNA genes. That is a lot. That is a lot of sequence. And those targeted 18,905 protein coding genes, usually around the transcription start site. 11,000 non-coding regions were used as controls, so this is very robust screen, let's say, and approximately each gene had about 5 to 10 single guide RNAs. So you know that this shit is legit, okay? So then the strains. So we have the screen, we have the cancer, we have the single guide RNAs, we have the library, everything is ready. Now we have to decide about the cells that you're going to use, right? And they had 2 different cells that they generated. So LNCap is a MCRPC cell line, but then they add the I. The I means that they actually added the plasmid with the crab and dead Cas9. And you have the other cell line, which is a little bit more aggressive, but they prioritized this one. Do you guys remember why? No? Come on, guys. Okay, I'll give you the answer.
It was from the start of the lecture, Matt.
Sorry?
It was from the start of the lecture, Matt.
The start of the lecture?
Yes, this was mentioned before.
The LNCap I?
Yeah.
Oh, because you came in when I was talking about but in the limitation.
I don't know, I'm just getting a sense of deja vu.
I was like, when did I say this? Okay, so the LNCap, yep. Okay, no. Exactly, that's the only thing. They just focused on the androgen-sensitive one because they're trying to find within the androgen-sensitive context what are the genes that are non-androgen dependent, okay? They could have worked with this one too, but probably their data set for this one was much better, so they made it look like they focused on this, okay? And so then what you do with this, one thing that was weak in their experiments is they had 2 replicates. The 2 replicates are, usually you try to get 3 and for each one of the cell line, not 2 replicates. Or even 3 is like weak, okay? But I guess in this case here, because the experiment is huge, the library of 200,000 single guide RNAs, 2 replicates is kind of acceptable, okay? One thing that is interesting, you're infecting the cells. Imagine infecting 200,000 different cells, right? And so it will be very hard to grab each piece and then extract the single guide RNAs in there and then sequence it and then compare all the data sets. But look, interestingly, in this kind of project, you're able to do it all together because of the sequencing approach, right? So this is very, very robust. It allows you to do this kind of big study and not think about which cell got infected with what, as long as you just pull everything together, okay? The actual best approach for this would be to separate one cell in each well and each cell be infected with one virus, and then from there do single cell RNA-seq. But it's way too much work. And there's one team that will be talking about single cell RNA-seq. So maybe you can refer to this. Anyways, so the next step is, what do you measure, right? So now you have your cells infected with those plasmids that have the single guide RNAs and all that. Then you have to make sure that you're looking for the single guide RNAs that decrease, right? Not increase. Why? Any idea? Yeah? Exactly. So if you're trying to find something that causes cancer, and this is a CRISPR-I screen, that means the CRISPR-I screen will inhibit that gene. By repressing that gene, then you kind of affect its ability to proliferate, so you'll end up decreasing the amount of cells. So when you decrease the amount of cells, you end up with less single guide RNA in your extract. If you were doing a CRISPR-A screen, and then you were targeting genes that allow for proliferation, then you would have more single guide RNA because you enhanced its ability to proliferate, because it activated even more of the genes that cause cancer. Make sense? Okay. Let me write the names a little bit because there's a few people. Who else? Sarah? No? Julian, Matthew. That's it? Alex? Yeah. And Akil. Okay, more controls. You could have talked about this control too. Was this graph easy to understand or not really? Kind of? Okay. So they took the 2 replicates that they had for the infected cells at T8, and they created this kind of average phenotype score, which tells you if the cells are dying or not, let's say. And they just did a correlation test on them, and you see that the negative control, which targets genes that are non-coding regions, center in the middle around 0 and 0. So that means they did not really affect the phenotype, okay? And it makes sense because these are non-coding regions, so why would they affect the proliferation ability of a cell? Now, one interesting thing here is that they filtered this data and they allowed for an expression level change of like 2-fold. This is something that's interesting because based on what you change in there, you can have more stringent data or less stringent data, and then you can have more false positives, things like this, right? So if they went, for example, a 5-fold change, then only these will be included, right? And maybe their answer would have changed, okay? But depending on the paper that you're working with, usually it goes from 1.5 to 2. Some fishy papers go below that because they don't see a phenotype and they're trying to include more data, okay? So then in this case here, it shows you that a lot of the genes there are a few genes that are contributing to the phenotype between the 2 replicates. And you're usually interested into these ones here. The reason why you don't see a lot of genes in the positive side, it's because this is a CRISPR-I screen. Right? So if it was a CRISPR-A screen, this region would be empty and this one would be full. Okay? Next, the second graph, the other graph that was shown, and this is another just example of a place where you could have actually shown some statistics. So when you're talking about comparing different data sets between them, you need to use statistics to make sure that those samples are those data sets are comparable. You cannot take a sample that was done by someone a few years ago using some sort of technique, and then a different data set using a different technique, and then comparing them just like that, because the values have no meaning if you move them from here to there, right? So you need to do these statistical tests that kind of turn things into comparable data sets. One of them is this Wilcox rank sum test, or the Mann-Whitney test, just different names for it. This was one important test that you could have potentially talked about. Obviously, you guys don't know all the statistical tests. I don't even know them, but t-test, probably we all do know it, right? Right? So if you see Mann-Whitney, maybe it's the one that you should be focusing on, right? Because this is the one that people are not aware of. But obviously for the statistical tests and for the limitations, I'm ignoring, okay? I'm giving you bonus points if you have them. If you don't, it's okay. But for example, for this paper that you have to submit, t-test, not interesting. We all know what t-test is, right? The idea here is to learn. So maybe when you find a weird test in there, that's the one you should potentially talk about, because it has a purpose other than just comparing 2 points together. But some people talked about the p-values, things like this. So yeah, it makes sense. You can include them. But actually, if you go in the paper and you go down at the bottom, you'll see statistical tests. Most of the thing they talk about is those weird tests, not the t-test. This graph here shows you that after some filtering, they got 1,472 genes that were differentially expressed. Of those, they show some controls here. So I'll just get to the controls right away. So the highlighted genes in this graph here are showing you these genes here, with most of them being essential for just regular cell activity. Related to cell division, right? So these are maybe not interesting in this case. One of them that comes up is androgen receptor. If you're doing a study about androgen-dependent and independent cancers, this needs to be your top hit, right? So this is another control to show that their kind of pipeline is structured. So you see how I'm talking about their data, right? Try to do this a little bit in your presentations, right? So that you kind of show that you understand the controls that are being used. So in this case here, I'm telling you that if you're doing work on AR and AR independent, this must be your top hit. If you don't have this, for example, and you try to publish a paper with a certain journal, they will tell you, okay, but you don't have even AR, so why are you talking about androgen dependent and independent? Maybe your data is actually misleading. You're expecting this to be your top. And they use it throughout the paper multiple times to talk about that. Okay. I don't know if I will have to finish all this. I'm going to talk just about KIF4A, kinesin factor 4A. But very quickly, they had to filter the data because most of these, you cannot just take 1,472 genes and say these are all independent. Some of them are independent, but they're unrelated to the cancer itself. They're just, cell division proteins or whatever. So then they did some filtering, clinical and functional. Again, when they do the clinical, you see that AR comes up as the top. Okay, let me just skip this because I already said this. But the AR comes up as the top hit, again confirming that their stuff is working. The other stuff, most of them seem to be unrelated to the cancer. So this is where you need to do your kind of research by yourself. Let's say if you're working in a lab, this gene always appears. Ribosome maturation factor is needed for everything, so it doesn't matter. Mitochondrial function, well, mitochondrial function is kind of needed everywhere. Ribosomal proteins, So mitochondrial ribosomal proteins, they're always there. One of them that is interesting is chromosome. So when you see chromosome segregation, you have to think, is it linked to all chromosome segregation or just this specific cancer, right? And for that, you have to kind of evaluate it using other data sets and things like this. In this case here, the only one that is specific to prostate cancer is the androgen receptor. But if you search the literature, you find that KIF4A has not been investigated in this sense, in the context of prostate cancer. So then maybe you want to focus on this one, right? But they did more filtering. They took a look at PICOS and DepMap, which are just databases that show you different things. In your paper, you could have talked about these in depth or just one or two sentences. That's all right. But these databases basically link your data. You can use them to find what has been found in clinical samples and in other CRISPR-I screens. DepMap tells you if that cancer is dependent on a specific protein or gene, and PICOS just gives you, so it's perturbation-induced CRISPR-I, whatever, database, and it just tells you if that gene is essential for that cancer, okay? So you need clinical data to then filter that out, and the idea is just to remove all the genes that have been found to cause poor growth, so low cell proliferation, but they act the same way everywhere else, in breast cancer, in normal cells, and so you want to filter those out, right? And maybe other screens found those genes, so you want to figure out if, maybe that gene that you see enriched or depleted is actually specific to just prostate or to prostate and breast cancer, for example, etc. When you do that, you end up with a new list. AR comes back as the top again, so that's a very strong control. HOXB13 is a gene that is known for breast cancer, prostate cancer, and actually our lab studies both AR, HOXB13, and KIF4A, okay? And then you have this WDR62, which is new. Actually, let me go here, which we are not sure what the hell it does. HOXB13 has already been implicated in prostate cancer. Another mitochondrial ribosomal protein. Usually when you see these, you ignore them, unless you're studying them directly. And then you have these two. The group decided to focus on this here because it's kind of novel. This one here seems to be a general use gene, so they kind of ignored it, okay? What else? I'm going very fast here. So KIF4A has been seen in other cancers, so obviously it's of interest, but not prostate cancer, so maybe it's your luck and it's KIF4A that's androgen dependent. And WDR62 has not been seen in other cancers, in prostate cancer, and so this one is also of interest. So this is where you kind of do filter to get the top hits, but then you have to do your own research from that point. Okay, a few graphs. They decided to focus on KIF4A because 33% of the MCRPC cases had it amplified. It's amplified because it's on the same arm as the AR receptor, so obviously it's of interest. And we're trying to look at something that is independent of androgen. So they did some comparison using the experiment plot to see if KIF4A expression was dependent on AR or not, and you can see that it is not. If it was, it would be kind of more increasing like this, right? So this is one that is very interesting because it's independent in cancer. It's one of the, in prostate cancer, of AR, and it's found in 33% of the cases. So this is highly of interest. And then if you look at these Mayer curves, you'll find that if you have high levels of KIF4A, then you end up being at risk of, your overall survival decreases basically. And then you can talk about the hazard ratio, which is a statistical way of thinking about this. Some people did in the assay, good stuff. But the idea is it's found clinically, amplified in 33% of the cases. On top of that, it shows a curve that has overall survivability that is lower. and seems to be independent of androgen. This is exactly what they're looking for. So that's why they focused on it. And then if you do inhibit its expression, then you end up, when doing those colony assays that tell you if there's proliferation or not, when you inhibit it using your single guide RNAs, then you see that there is a decrease of colonies produced. That means that you're actually affecting the cell cycle or the proliferation ability of this cancer. If you overexpress it, you see the opposite. And they did this in multiple different cell lines. Obviously, the ones that are interesting, very aggressive, 22RV1, C42, and LNCAP. So this is some good data. Then they also looked at migration and invasion assays. Metastasis requires you to kind of remove yourself from the environment and then start walking and then go somewhere else. So this is the migration and invasion assay. They saw that overexpression of the KIF4A leads to a increase in, it's called anchorage independent cell growth. That means they don't depend, because cells, for them to grow, they're required to be in contact with other things. And so if there's no contact, they will actually start moving. And so this is called anchorage independent cell growth, which is another way of saying metastasis. And so you can see that overexpression of KIF4A leads to an increase in migration, and a knockdown leads to decrease in migration. Seems to be the perfect gene, right? Now you have to test it in vivo. To test it in vivo, many ways of doing this. They went with the cheapest way, let's say, the least relevant way, which is to create your own cells and then put them in the mice. Inject them with a syringe, leave them there for a few days, and then you'll see tumors to start growing. Okay. Now they did something additional to that. Their CRISPR system that they used to express KIF4A or downregulate it required an inducible promoter that reacted to doxycycline. So then you will inject the mice with doxycycline or not, and if you inject it, you will activate this single guide RNA. And you can see that when you activate the single guide RNA, you do decrease the size of the tumors. Okay? Obviously, I talked about the limitations of this, right? I will go back to it very quickly. The next step is to actually characterize KIF4A. Now, I will go very quickly through this. Kinesin, if you remember, you've probably seen this video. It's a motor protein that carries some sort of load on the microtubules. And so this is the known function of it. So obviously, if you play with it, you'll probably play with the cell cycle, because it transports many things, including chromosomes and things like this. Okay. One thing that they did is that they looked at other functions of KIF4A using these gene set enrichment analysis, GSEA. And they found that one thing that popped up, one of the Go terms that popped up the highest when you played with the level of KIF4A is DNA methylation. This is a novel role that was not kind of known for KIF4A. So you have control and knockdown of KIF4A. You compare them in terms of protein, in terms of transcripts that are there, using RNA-seq or whatever, and you'll see that one of the Go terms that appears suddenly is DNA methylation. And this has a big role in cancer. Then they used a technique to see how KIF4A affected DNA methylation. They did something called ATAC-seq. ATAC-seq basically just cuts your DNA at open places in the chromatin, so it tells you which regions are active. Since we're studying DNA methylation, then we need to study open regions of the chromatin. It will cut it into smaller pieces and ligate adapters to it, and then you can just sequence it. And when you sequence it, you'll find which regions are open, and that tells you what is the state of the DNA and transcriptional state of the cell at that specific moment. And that can tell you about the downstream effectors of KIF4A that lead to causing this prostate cancer. Okay, so there's just additional stuff. ATAC-seq is a very cool technique. It's quite recent. So this is maybe the other technique that I would have expected you to talk about because people don't know it, but obviously not everyone talked about this part of the, usually you stopped at identified the 2 genes and then did some mouse work, most of the people. So I was not expecting you to get into in depth for this. Wow. Very fast. So obviously for your presentations, don't include a lot of data like this, okay? Because if you go through the paper, you will go beyond the 20 minutes. My idea, how I work is, I don't expect you to present everything, but I expect you to present 1 or 2 things very well, right? So if it was for me presenting, I would have probably skipped most of it, just talked about the screen, directly give the results of the filtering, and then talk about the mice work for one gene. No need to talk about 2, because we could go through the whole list of genes, right? So if you're not sure about the amount that you need to include, you can ask me by email for your paper, and then I'll look at it and tell you, okay? Last question, which is the part of the discussion. So instead of going through the discussion and saying they did this, they did that, and that is limited and that is good, I'm going to cover here just the part that is future perspectives, let's say, okay? And then in the next articles I'm going to present, I will focus more on the other aspects of the discussion. So do you think that those 2 factors they found could be potential clinical targets for prostate cancer? Anybody? Maybe.
Well, after the study has been resolved, we do see that there are some things we could be trying out. There have been some key genes that have been identified.
Mm-hmm.
We try disabling, enabling, playing around with that, and see how that alters the functionality of the gene. The whole difficult part is actually landing the drug so that it goes over to that gene and disables it or fixates itself to it or disables its product like its enzyme.
Yeah. Alessandra, do you?
I don't know what I can add, but since they found that the genes are specific to prostate cancer, I guess it's a good starting point to further research. But obviously, like from now until a clinical program is developed, could be like years or maybe a decade.
What's missing? Yeah, Matthew.
They know it's associated with prostate cancer, but they don't know how it works or why
It does.
it's actually doing.
Yeah. So few things that come up. The mechanism is unknown. We know that that protein is there. It's associated with the cancer, but we don't know what the hell it does, right? So we need to figure out what it does, because obviously if it is in the prostate cancer, but it's kind of necessary for everything else, you cannot use it as a target, right? So usually the way you go about this, before getting to the clinical trial, you need to do more things from here. Do you guys have an idea of what you need to do? So you know that it's found in prostate cancer, but how do you make sure that it's fit for a clinical program? Yeah.
You have to do more sequencing with another clinical cohort.
Mm-hmm.
Can you check exactly for these genes?
Yep. Okay.
I would test to see if, like, knockdown of that gene in normal, regular cells doesn't result in immediate death, so that if there are any off-target effects, not lethal or whatever.
Yeah. Alex.
It's kind of similar to what he was saying. I was just going to say, look at the expression levels of
Healthy.
and other tissues.
Yeah. Okay. One thing. Yeah.
Maybe we can just check with single cell RNA that we're going to use, like we can get like this profiles of all genes and find these pathways. Maybe it's just not that one pathway, maybe it's like the other one down-regulating another thing too.
Yeah. Usually when you have factors this big, you try to find something much lower in the chain so that you don't destroy everything. So developing drugs for something is kind of hard to do. So you need to develop the drug for it. You need to put those cells back in the mice. You need to try to treat those with that drug and see what happens to the mice. When you know that that is okay, then you need to understand the mechanism for it, because now you have a phenotype, but you don't want necessarily for the cancer to come back in 5 years and be even worse, right? So you have to kind of make sure that your drug is killing those cells, but not destroying the organism as well. And one thing that has to happen before the clinical trials is you need to put it into the microenvironment that those cells are found in. And this is quite hard to do. You usually try to do organoids for this, or try to kind of have the cancer happen naturally, and then instead of injecting the cells, you induce the cancer naturally by giving too much testosterone or things like this. And then the mice just end up killing each other. They actually bite each other, and like, it's crazy. So it's very hard to do. And then maybe you can justify a clinical program, but I looked online just to see if there were any clinical programs that start from this. There are none, so you can find that online. So probably something happened en cours de route to the clinical program. Okay? So that's it for me. Do you guys have any questions?